All right, let's jump right into this explainer. I'll be stepping into the role of your professor today, and together we're going to explore the absolutely fascinating biological disassembly line of animal nutrition. We're talking about how nature engineered some of the most complex, efficient, and honestly awe-inspiring biological machinery on the entire planet. Yep, we are diving deep into the evolutionary journey of the digestive system.
Here's our syllabus for today's class. We'll cover 1. The essential nutrients, 2. The evolution of digestive compartments, 3. The alimentary canal, 4. Inside the mammalian stomach, and 5. The perfection of digestion.
Okay, kicking things off with Section 1, the essential nutrients. So, we start with a really fundamental biological problem. How do we get the fuel and raw materials our bodies desperately need but just can't create from scratch? I want you to ask yourself this foundational question. Why do animals, unlike plants, have to constantly devour other organisms to fuel their cellular replication and growth? Think about it for a second. A plant can just sit there in the sun, pull carbon dioxide from the air, and literally build itself. We definitely can't do that. To survive and reproduce, we have to carefully balance our consumption, storage, and use of food. We need that chemical energy to produce ATP, and we need raw organic carbon and nitrogen to actually build our own tissues.
But beyond just basic fuel, the answer to our question really lies in essential nutrients. These act as mandatory substrates and coenzymes that our bodies simply cannot manufacture, mainly because we lack the evolutionary genetic blueprints to do so. In other words, even if we had all the carbon and nitrogen in the world, our cellular factories are missing a few absolutely crucial assembly machines. And because we lack those specific enzymes, we're forced to import the finished products directly from our diet. There's a quote that brilliantly illustrates this exact concept. Albert Szent-Györgyi, the guy who actually discovered vitamin C, once quipped, "A vitamin is a substance that makes you ill if you don't eat it." It's so true, right? Relying on external sources for our chemical building blocks leaves us incredibly vulnerable if our diets happen to be incomplete. If you don't consume enough vitamin C, for instance, your body can't synthesize collagen and you develop scurvy. You literally begin to fall apart just because of one missing environmental input. Broadly speaking, there are 4 main classes of these mandatory imports. We've got essential amino acids, essential fatty acids, vitamins, and minerals. Just to put this into perspective, out of the 20 standard amino acids required to make proteins, our bodies can only synthesize about half of them. That means the remaining essential amino acids must be violently cleaved from the proteins of the food we eat. We literally take the proteins of another organism, break them all the way down into their base amino acids, and then use those borrowed pieces to build human proteins. Moving right along to Section 2, evolution of digestive compartments. Now this brings us to a frankly terrifying biological paradox. How exactly do we utilize destructive protein-cleaving enzymes without our stomach just dissolving its own cellular walls? Remember, the enzymes that hydrolyze the proteins and fats in your food are perfectly capable of hydrolyzing the proteins and fats that make up your own tissues. Unchecked, digestion is basically self-destruction. Well, evolution solved this massive problem through compartmentalization. The earliest and simplest method was intracellular digestion. Picture a single cell engulfing a tiny food particle to form a food vacuole. That vacuole then fuses with a lysosome, which is just a tiny cellular packet of enzymes. The digestion happens safely inside that sealed microscopic bubble. Believe it or not, sponges still do this today. But if you want to be a larger animal and eat larger prey, evolution had to transition to extracellular digestion. This is the breakdown of food in compartments that are continuous with the outside of the animal's body. One of the very first evolutionary models for this is the gastrovascular cavity. Early carnivorous predators, like the microscopic freshwater hydra, use a gastrovascular cavity to stuff prey through a single opening, digest it, and then expel the waste back out that exact same door. Yep, the single opening serves as both the mouth and the anus. Gland cells secrete enzymes into this central pouch, breaking down the prey so the surrounding cells can safely absorb the nutrients. Which perfectly segues into Section 3, the alimentary canal. There is a massive evolutionary advantage when we contrast the old batch processing of early organisms with the sequential efficiency of the alimentary canal. Think about it. If you have a gastrovascular cavity, you literally have to wait until you are entirely finished digesting and expelling a meal before you can even think about eating again. But with a complete digestive tract, an alimentary canal with 2 distinct openings, you can continually process food. You can ingest a brand new meal while the old one is still being digested further down the line.
By moving food in a single direction, the alimentary canal allows different compartments to specialize drastically. It basically turns the entire digestive tract into a highly efficient multi-stage disassembly line. The mouth handles the mechanical breakdown and early carbohydrate digestion. The stomach handles intense protein digestion. The small intestine tackles the bulk of chemical hydrolysis and nutrient absorption. And finally, the large intestine recovers water. It's essentially an assembly line running in reverse.
Let's zoom in a bit for Section 4, inside the mammalian stomach.
Mechanically speaking, the stomach acts just like an industrial concrete mixer. Its walls are lined with smooth muscle that relentlessly contracts, completely pulverizing your meal and blending it with gastric juices every 20 seconds. This rigorous churning takes what was once a recognizable bite of food and entirely reduces it to a nutrient-rich, highly acidic broth known as chyme.
Chemically, this environment is shockingly hostile. The gastric juice in your stomach sits at a pH level of 2. To put that into perspective, that is literally acidic enough to dissolve iron nails. No joke. And it effectively kills off most of the bacteria you ingest. Biologically, this super low pH denatures or unfolds the tightly bundled proteins in your food, exposing their peptide bonds so that digestive enzymes can actually get to work.
But hold on, remember our paradox? How do we not digest our own stomach? The crucial takeaway here is that our cells protect themselves by secreting inactive precursors. Let's walk through the step-by-step process. First, parietal cells pump hydrogen and chloride ions separately into the stomach cavity. They only combine to form hydrochloric acid once they are safely in the lumen. Meanwhile, chief cells secrete an inactive protein called pepsinogen. At this stage, it's totally harmless. But when pepsinogen hits that extremely acidic environment, the acid clips a piece off, weaponizing it into the active protein-cleaving enzyme called pepsin. It's wild that chemistry only arms the bomb once it's safely outside the cells that build it.
And finally, Section 5, the perfection of digestion. The digestive system really is a masterclass in form-fitting function. Depending on whether an animal tears flesh or grinds leaves, evolution stretches, expands, and completely modifies this digestive tubing. Carnivores, who might go days between kills, have massive, expandable stomachs. A 200-kilogram African lion can consume 40 kilograms of meat in a single sitting. Herbivores, on the other hand, eat plant matter rich in cellulose, which is incredibly difficult to digest. So they have significantly longer alimentary canals to provide way more time for digestion. And crucially, they invite trillions of mutualistic bacteria into specialized fermentation chambers in their gut to act as a supplementary workforce, breaking down cellulose into usable sugars. We literally rely on the genetics of other organisms to digest our food for us. And that brings us to the most incredible realization of all. Our digestive systems are not static. They are deeply, inextricably tied to our environment. We've seen how our anatomy shifted to process meat, to host microbiomes, and to efficiently extract the essential building blocks of life. Given the profound impact our diet has on our anatomy and our symbiotic microbiome, I want to leave you with this final thought. As we consume increasingly processed foods and drastically alter our microbial environments, how might our own evolutionary journey of digestion adapt to the foods of our future? Thanks so much for joining me in this explainer, and keep asking those big questions.